Hello, welcome to the Sahoo Lab. We are located in the Rutgers Newark campus in the Department of Biological Sciences. We are a molecular neurobiology lab. The main focus of our lab is to understand how neurons develop and how do they repair themselves after the injury. Then we also seek to understand how can we harness this knowledge to gain functional recovery.
We are a diverse and curious team of postdocs, graduate students, and undergraduates. Together, we combine molecular biology, imaging, and bioinformatics to uncover how neurons respond to signals, stress, and injury. To answer these questions, we use a variety of model systems. These include culture neurons and rodent models of sciatic nerve injury and traumatic brain injury. A central question that we ask in the lab is how mRNA translation in the neurons is regulated. One clue lies in local mRNA translation. It is the ability of neurons to make proteins right where they are needed, far from the cell body. We study how this process is regulated by specialized RNA protein complexes called as the stress granules. These are dynamic structures that are generally known to be formed during cellular stress. However, we find that neurons utilize stress granules under physiological conditions to store non-translating mRNAs. In particular, we focus on G3BP1 positive stress granules, which control whether an mRNA is translated or stored. Our team uses cutting-edge molecular, cellular, and genetic tools to map signaling pathways that control neuronal development and regeneration. Science in our lab is a team effort. We take pride in mentoring young scientists, fostering curiosity, and building an inclusive, supportive environment. Every student, from undergraduate to postdoc, contributes to advancing our collective mission. We are the Sahoo Lab, decoding the molecular mechanisms that drive neuronal growth, repair, and regeneration. Together, we seek to understand how neurons develop and how injured neurons reconnect and restore function.